Welcome, everyone, to today's Explainer. I'm absolutely thrilled to be your guest lecturer today as we tackle a really high-yield synthesis of advanced bacterial anatomy and physiology. Now, for those of you at the postgraduate level, you already know that simply memorizing a list of cellular structures, well, that's just not going to cut it anymore. We need to connect these physical architectures directly to clinical pathogenesis, immune evasion, and modern pharmacology. To make sure we hit all those marks, here is our roadmap for today. We'll be going through 1. The outer bacterial defenses, 2. The cell wall dichotomy, 3. Membranes and surface appendages, 4. The internal genetic machinery, 5. Ribosomes and metabolic structures, and lastly, 6. Bacterial spores and survival.
Okay, let's dive into this. We are taking a spatial journey today, navigating this organism exactly from the outside in. Think of it like mapping out the precise path an antimicrobial drug or even one of our own host immune cells has to take to breach the organism. From the outermost protective shields all the way down into the metabolic engine and the genetic core, every single structure is either a brilliant evolutionary barrier or a prime clinical target. So let's start our approach right at the very edge. Section 1. The outer bacterial defenses. Our very first major barrier is the capsule, or the slime layer. This is typically a thick, gelatinous layer covering the entire bacterium, and it's generally made up of complex polysaccharide structures. But since you're at the postgraduate level, you are definitely expected to know the critical clinical exception here. The capsule of Bacillus anthracis is actually composed of polymerized D-glutamic acid. It's this unique polypeptide armor that plays a massive role in the lethal virulence of anthrax. So why do we actually care so much about the capsule on the warts? Because honestly, these outer defenses are formidable. The capsule literally inhibits our own immune phagocytes from ingesting and killing the bacteria, and it blocks host lysosomes. On top of that, it enables the growth of biofilms. When a bacterium sticks to an inert clinical surface, think about an IV catheter, a prosthetic heart valve, or even your teeth. That biofilm creates a microscopic fortress. It completely blocks out host cells and antibiotics, which is exactly what drives those incredibly stubborn nosocomial hospital infections. But on the flip side, it's also exactly why we harvest these specific capsular structures to use as potent antigens in our vaccines, like the pneumococcal vaccine. Section 2. The cell wall dichotomy. Moving just inward from the capsule, we have to define the fundamental brick and mortar of the wall itself. I'm talking about peptidoglycan, also known as murine. This is the main structural polymer of the bacterial cell wall. It's built from these interlocking chains of two alternating sugars, N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM. Grasping this specific polymer is absolutely crucial. It's the foundation for understanding both how gram staining works in the lab and how beta-lactam pharmacology works in your patients. This brilliantly illustrates that architectural strength. You can clearly see the overlapping NAG and NAM molecules running vertically, and they are cross-linked horizontally by those critical peptide interbridges. It essentially creates this tight, mesh-like exoskeleton all the way around the cell. Now, this is present in almost all bacteria, but again, file away this notable clinical exception. Organisms like mycoplasma and ureaplasma entirely lack this wall. And now we face what is arguably the most critical comparative dichotomy in microbiology. We've got the gram-positive cell wall and the gram-negative cell wall. Notice the stark visual difference here. The gram-positive wall relies on the sheer massive thickness of peptidoglycan to defend itself. The gram-negative envelope, however, relies on a highly complex, multi-layered fortress. Let's break those differences down. A gram-positive bacterium cell wall is made up of 50 to 90% thick peptidoglycan. Importantly, embedded right in there are teichoic and lipoteichoic acids, and those heavily promote the cell's ability to stick to host epithelial cells. Conversely, look at the gram-negative wall. It has a remarkably thin peptidoglycan layer, just 5 to 10% of the wall material, but it makes up for that structural vulnerability with an additional outer membrane, a periplasmic space that's just packed with digestive enzymes and transport proteins, and specific porin channels that strictly regulate what gets in and out of the cell. So the crucial point here is the extreme clinical danger presented by that gram-negative envelope. That outer membrane contains a structure called lipopolysaccharide, or LPS, and embedded deep within that LPS is lipid A. Lipid A is a potent endotoxin.
When a gram-negative bacterium is destroyed, whether by our immune system or by the antibiotics we administer, this lipid A is dumped right into the host's bloodstream. That triggers a massive, uncontrolled inflammatory cascade, which is responsible for terrifying endotoxic activities like high fever, severe hypotension, and life-threatening septicemia. To wrap up our look at this structural boundary, you have to remember that the cell wall is essentially the ultimate pharmacological target. It maintains the cell's shape, it protects the organism from catastrophic osmotic lysis, it dictates our gram-stain reactivity, and it heavily influences pathogenicity. Most importantly for you as future clinicians, because it is so incredibly vital for bacterial survival, it is the primary site of action for our heavy-hitting antimicrobial agents, like penicillins and cephalosporins. Those drugs work specifically by inhibiting those peptidoglycan crosslinks we were just looking at. Section 3. Membranes and surface appendages. Okay, having breached that rigid wall, sitting immediately beneath it is the plasma, or cytoplasmic membrane. It acts as a strict, semi-permeable boundary separating the wall from the internal cytoplasm. It's composed mostly of lipoproteins with just a few carbohydrates, and it's the main site for active transport and for synthesizing those cell wall precursors. There's a key structural distinction here that always pops up on exams. Unlike our human eukaryotic cells, bacterial plasma membranes generally do not contain sterols. Again, the one major exception being mycoplasma. Before we plunge completely into the cytoplasm, we really need to acknowledge the physical appendages reaching out from this membrane surface. We have pili. These are the shorter, straighter, hair-like projections made of a protein called pyrin. Common pili or fimbriae help the bacteria attach to host cells, while the longer sex pili are involved in conjugation. Contrast that with flagella. These are the long, filamentous appendages made of flagellin. They act like highly antigenic outboard motors, driving bacterial motility. They also carry vital antigenic determinants, like the H antigens you find on gram-negative enteric bacteria. Now, mentioning conjugation brings up a really fascinating biological and, quite frankly, clinical problem. If bacterial cells lack a membrane-bound nucleus to carefully organize their genetics, how on earth do they manage to so rapidly transfer resistance mechanisms to one another and survive our best antibiotics? Section 4. The internal genetic machinery. The answer to that question lies deep within the cytoplasm. While the main circular bacterial chromosome just sort of floats in a region called the nucleoid, the real agents of clinical chaos are the plasmids. These are extrachromosomal, double-stranded DNA molecules. They can replicate totally on their own, and crucially, they can be easily passed from bacterium to bacterium right through those sex pili we just talked about. Make no mistake, plasmids are the primary engine behind the modern crisis of antibiotic resistance. They directly encode the genes for that resistance, as well as major pathogenesis factors like devastating enzymes and toxins. Section 5. Ribosomes and metabolic structures. Now, what's really interesting about this next part is how we exploit their machinery. Now that we've seen the genetic instructions, let's look at the cellular factories that translate that code into physical action, the ribosomes. These structures are made of RNA and proteins. Notice the exact mathematical sizing here. The bacterial ribosome is exactly 70S in size, made up of a larger 50S subunit and a smaller 30S subunit. This is a massive exploitable structural discrepancy from our own human eukaryotic ribosomes, which are larger, sitting at 80S. Because of this precise evolutionary difference, we can deploy antimicrobials that specifically recognize, bind to, and jam up the 70S bacterial ribosome. That shuts down bacterial protein synthesis without harming our own human cells. Floating around those ribosomes in the cytoplasm are a few other metabolic structures worth noting. Inclusion, or nutrient granules, serve as these little storage vaults for energy. For instance, you'll characteristically see volutin granules in Corynebacterium species, where they store essential nutrients. We also see mesosomes. These look like convoluted indentations of the cytoplasmic membrane itself. They act as central hubs for respiratory enzyme activity, and they also help tightly coordinate nuclear and cytoplasmic division during binary fission. Section 6. Bacterial spores and survival. Let's move to and see how this all builds into the ultimate, desperate survival mechanism of a bacterium facing a hostile world. When standard anatomy is no longer enough to survive a barren environment, the bacterium forms an endospore. Certain highly dangerous bacteria, primarily those within the Clostridia and Bacillus genera, can form these dense, rounded structures inside the parent cell when conditions get tough. And honestly, endospores are biologically terrifying. Once formed, they drop to absolutely zero metabolic activity. they become virtually indestructible. They resist extreme heat, lethal radiation, and severe drying out. They can just sit there, completely dormant, in the soil or on a hospital surface for literally hundreds of years, just waiting patiently for the exact right host to come along.
So I want to leave you with this thought. It's a profound, almost philosophical challenge for modern medicine. If a single dormant endospore can outlast centuries of desiccation and radiation, only to violently reactivate the second it's swallowed by a host, how can our pharmacology ever truly permanently win this evolutionary arms race? It's a formidable question for sure, and it's one you absolutely must carry with you into the clinic. Thank you so much for joining me on this structural journey today. Keep studying, and I will see you in the next explainer.